Hello everyone, welcome back to Genomics for All, episode number 11. It's been an amazing experience so far and we will continue this. So today I'll be talking about genomics for infectious diseases. In particular, as you all know, India has a big problem of infectious diseases, and COVID-19, as we all know, was the biggest infectious disease of this century. So today we will be talking of another infectious disease which is very, very important for India, particularly that's tuberculosis. And we will have this series, we'll continue this with many other infectious diseases like human papilloma virus and many other things that we do at MyBiome. So to put things into a perspective, tuberculosis worldwide, 10 million people fall ill with tuberculosis every year, and 1.5 million people die of this disease every year. In India, the numbers are big, like 1/4 of this whole number is in India itself. So around 25 lakh people get TB, and around 2.5 lakh people die of TB. That's about 10% morbidity rate, which is very, very high. It's higher than any other infectious disease, even higher than COVID mortality rate. So that is what the numbers say. These numbers are alarming, very something to worry about. Why a big country like India with 1.4 billion people, we have so many cases of TB even now. We thought infectious diseases are something of the past. TB is something of the past, but it is not. Now, where is the problem? First of all, TB treatments, governments have done a lot, I would say. Government of India has done a lot on TB program, giving medications, giving nutrition, supplements, etc. But then came the drug resistance problem, which everyone is around the world is tackling this menace. It's a next pandemic, I would say. The drug resistant TB, we have 15 drugs approved, endorsed by WHO, and now we have this TB which is sometimes resistant to all the 15 or 14 or 10 is normal in India. So that is where now the problem is. How do we deal with this drug resistance menace that is happening in tuberculosis, particularly for a country like India, where our densities are pretty high, right? If you see Mumbai, for example, the densities are very high, population densities. So we have to be very careful of the spread of drug-resistant TB and drug-resistant TB in particular. How do we tackle this problem? First is, before any treatment, we have to understand what drug resistance does a TB bacterium have. So there are many tests designed to understand this drug resistance problem, and tests like drug sensitivity test. We had tests like qPCR, RT-PCR, the famous RT-PCR that all of you know during COVID-19, line probe assay, or point of contact nucleic acid amplification tests. Many tests, LAMP, that was ligation mediated amplification, etc. So many tests came in, but then the entire comprehensive test to actually diagnose and determine the drug resistance of all 15 drugs endorsed by WHO was not available in all these tests. They were doing either 2 or the maximum was 10. So now came next generation sequencing. What is next generation sequencing? We have been talking about in our own series, Genomics for All, for a long time now. In the previous episodes, we defined what is next generation sequencing, etc. So I wouldn't go into the details of the technology, but next generation sequencing, as you all will now understand, is a powerful technology. And for drug resistance TB, there has been a lot of work happening in this space, as you can sequence the whole genome sequence of the TB organism, and then mark which of these genes that are responsible for the drug resistance of the TB are mutated, and that's why they are giving drug resistance. So it is always the case that some genes are mutated or changed in order to achieve drug resistance in TB. So now this is how it is done. We sequence the whole genome sequence of the bacterium. bioinformatically on the computer, we determine what are the changes that have happened and tell the doctor, the clinician, that these are the drugs that this bacterium will not respond to, and these are the drugs that it will respond to. So the doctor has a clear picture of which drugs to use, so that we don't overprescribe drugs. And over the counter is a bad practice that happens in India. So that is not allowed for TB, and there are strict laws covering that. So that's fortunate. We are fortunate about it. But overprescription of these 15 drugs, like giving all the 15 drugs to an individual, is also a big problem. So we define this. So this is where precision medicine, as we talked about in our previous episodes, has been applied to TB. Now, whole genome sequencing is a costly affair. It looks very simple, but then would Indian patients having TB, can they afford this? The answer is no. So we at MyBiome, now coming to what we do at MyBiome, have come up with a new solution wherein we took the targeted region of all these 15 drugs and made a small panel, very compact panel that covers only 15 drugs, 15 the genes for the 15 drugs. And this, we call it the targeted NGS, or targeted NGS kit that we have from MyBiome. So now this kit is better than whole genome sequencing in the sense that it is cheaper first, because it is a targeted NGS and not the whole genome sequence. It is faster because it can do directly now from sputum, which whole genome sequencing was difficult to do from sputum based DNA. And it is better because now you can do lot more higher coverage because it's a small panel in the same cost. So it's cheaper, better, faster, the targeted NGS panel that we have at MyBiome. And I encourage all of you to read more about our research, more about our new innovations in this area. We have a patent filed in this technology for the targeted NGS panel, and happy to discuss with any of you on this technology and more for genomics for infectious disease. Please stay tuned for more on genomics for many other infectious disease other than tuberculosis that are equally important for a country like India. Thank you. Thank you, everyone.